Hi, welcome back. What I want to do in this video is look at the biosynthesis of heme. And we're going to accomplish this in several videos, and I thought it'd be a good idea since we're getting into P450s and all sorts of hemes and stuff like that. So I thought it'd be fun to look at the biosynthesis of this molecule. And in this particular video, we're specifically going to look at the biosynthesis of δ-aminolevulinic. And we're just going to do this first reaction in this video, and it turns out this reaction is the committed step in the synthesis. So we're going to synthesize δ-aminolevulinic. And this is catalyzed by the enzyme δ-aminolevulinic acid synthase, right? Okay. Now, essentially what this enzyme is going to do, and I'll draw the initial substrates, it's going to take succinyl-CoA, right? It's going to take succinyl-CoA, right, out of the TCA cycle, and it's essentially going to condense it with glycine. It's essentially going to condense it with glycine. Okay? So, let's think about this, right? To make one δ-aminolevulinic acid, it's going to require one of each of these, right? It's going to take one of each of those to synthesize one δ-aminolevulinic. But it turns out that to make one heme, it takes 8 δ-aminolevulinic acids. So essentially to make one heme, it takes 8 succinyl-CoAs and 8 glycines. And you could imagine that we're going to have to take the succinyl-CoA out of the TCA cycle, and you can also imagine it's going to be a very energetically exhaustive process, right? So if I have succinyl-CoA, right, what's the enzyme in the TCA cycle that consumes it? Well, it's succinyl coenzyme A synthetase, right? Succinyl coenzyme A synthetase, and it ends up generating succinate, right? Well, what is ultimately produced in that reaction? Well, we put in a GDP, right? We put in a GDP, right? And we get out a GTP, right? Let's go a little further, right? If I use succinate dehydrogenase, right? Here's succinate dehydrogenase. What do I get out of that? Well, I ultimately get out an FADH2, right? And then, of course, I'm going to go to fumarate, right? Here's fumarate. And then I'm going to go to malate, right? And specifically the L isomer. And then what's going to happen? Well, I'm running out of room, but essentially I'm going to consume that with malate dehydrogenase. And what do I get out of that? Well, I get out an NADH. Right? I get out an NADH. So let's go and circle all the good things we would have normally got out of succinyl-CoA metabolism. Well, I got out a GTP, which will go form ATP, right? I get out an FADH2, right? And I get out an NADH, right? So let's think about this. If I take 8 to make one heme, if I take 8 succinyl-CoAs out of the TCA cycle, I end up losing, so I have lost 8 NADHs, right? I've lost 8 FADH2s, right? And I've also lost 8 ATPs. So this is a very energetically exhaustive process, right? You don't want to have to do this if you don't have to. So it would make sense for there to be some extensive regulation on this enzyme. And in fact, there is. In fact, δ-aminolevulinic acid synthase is an allosteric enzyme. It is an allosteric enzyme. But first, before we get into the regulation, let's actually look at the general reaction and actually they typically abbreviate this ALA, aminolevulinic acid. So let's look at ALA synthase. So the initial product that we're going to get something called alpha amino beta ketoadipate. So if we think about it, right, adipate is a 6-carbon diterminal carboxylate, right? So 1, 2, 3, 4, 5, 6, right? So here's adipate, right? And if it's alpha amino, right, here's going to be an amino. Here's my amino. And here is my beta keto, right? And if you think about it, look at it. And actually, in this particular reaction, we lost a coenzyme A, right? So that's this right here, right? And if we look at it, right, you can see the glycine right here, right? And then here, over here, is the succinyl group, right? So you can see the individual components still in the molecule, right? Right? And then this, and let me go ahead and write this. This is alpha amino beta keto adipate, alpha amino beta keto adipate. And this is just an intermediate. This is just an intermediate. It's actually unstable, and what we're going to find is that it gets decarboxylated by ALA synthase. So you lose a carbon dioxide. So what you end up with is this. You end up with this. So instead of being actually, let me erase that. Instead of being 6 carbons, now it's 5 carbons. So 1, 2, 3, 4, 5. There's my amine, right? There's my amine. And then I have my carbonyl right here. So this right here is aminolevulinic, right? And so to make one heme, I need 8 of these. So 8 aminolevulinates ultimately go to one heme, right? So ultimately, it's going to consume 8 succinyl-CoAs and 8 glycines. Okay. And like we mentioned, in order to, if you have an energy exhaustive process like this, and certainly this is very energetically exhaustive, you want to have some extensive regulation on it, and in fact you do. And the first form of regulation is really just the synthesis of the enzyme. So it turns out that the enzyme has a fairly short half-life. I'm not exactly sure what it is, but the enzyme has a short half-life. Let me do it like this. A short half-life, right? It has a short half-life. And so a lot of the synthesis of ALA is controlled by the synthesis of the enzyme. And there are generally two molecules that speed up the synthesis. One of them are barbiturates. Barbiturates generally look like this. Let me get some space. Barbiturates generally look like this. They generally look like this. So here is one barbiturate, and you have 2 R groups right here. So this is a barbiturate derivative. If these were both hydrogens, if both of these R groups were hydrogens, this would be barbituric acid. But barbiturates tend to speed up the synthesis of ALA. And actually, there's another molecule that does this, a molecule you may be more familiar with, and that is and actually it's a class of molecules, but it's any steroid that has a double bond at carbon number 4. So I'm drawing an example here, and here's an example of one that has a double bond at carbon number 4. And so when we're numbering the steroids, the one with the oxygen attaches carbon 3, so this is carbon 4. And so whenever you have a double bond at carbon 4, so we're specifically talking about a double bond at this position in the steroid. And actually, let me go ahead and do this. Well, let me do that. Actually, this is just an example of one that can do it. But specifically, the steroids that have a double bond at carbon number 4, and certainly this is androstenedione, and another example would be this as a hydroxyl group, right? And that would be testosterone. These molecules also increase the synthesis of aminolevulinic acid synthase. So if you increase the synthesis of the enzyme, there's going to be more ALA and therefore more heme. And because this enzyme is the committed step, once you form ALA is committed to forming heme. So that's why you want to have as much regulation on it as possible, because you're bypassing the production of all this energy, right? So if you're bypassing it, you want to have regulation on it. Okay. And so the first regulation is regulation of the synthesis of the enzyme. But also aminolevulinic acid synthase is an allosteric enzyme. And it turns out that it has a primary allosteric effector, and I'm going to draw it right here. And actually, this is actually the actual final product of the whole synthesis. And actually, I might also qualify this. It's thought to be an allosteric enzyme. I don't know that they've actually proved that it's allosteric, but it's thought to be regulated allosterically, and it's regulated allosterically by this molecule, and this molecule is heme. It's regulated by, and it's going to be heme. It takes a while to draw sometimes, but it's allosterically inhibited by heme. And really, this is sort of similar to a feedback mechanism, right? If you already have lots of heme, right? If you have lots of heme, you don't want the enzyme to be active. So heme will feed back, and it will allosterically inhibit ALA synthase, right? It will allosterically inhibit ALA synthase. So when you build up a lot of the final product, you don't need any more of it, so it shuts off. And actually, I want to come back here and look at this steroid. And actually, one thing about this is that these particular steroids that are like this are actually they're more prevalent in males, and so this is actually one of the reasons why males have higher concentrations of hemoglobin than females, because hemoglobin ultimately contains this heme in it, and delta-aminolevulinic acid is going to form heme. So there's a lot to learn about this particular enzyme, delta-aminolevulinic acid synthase, but what we ultimately form is delta-aminolevulinic acid. Okay, and this particular molecule is going to be consumed by porphobilinogen synthase, and that is going to kick off the rest of the synthesis of heme. And we'll look at that in the next video. See you soon.